So I'm going to start because this is a subject dear to my heart. So I'm going to start by a quick land acknowledgement for Canada. So I respectfully recognize and acknowledge the relationship that the First Nations, Inuit, and Métis people across Canada have with the land that all Canadians live on and enjoy. I recognize human rights of people who have suffered from systemic racism and discrimination in Canada, and I seek to provide a welcoming, engaging, and safe community for everyone. So thank you, everybody, for joining us today for the BINA AIMM user group. My name is Judith. I need to send regrets coming from Katrina. She's actively working on the final steps for a grant, which, if it gets funded, will have an important, significant, positive impact on automated image management and metadata annotation. So on that, I will... do you want to do the announcement first, Vanessa? Okay, so I'll let Vanessa go, and then we'll introduce our speakers today.
Sure, we'll do just some quick announcements just to share. We've got a BINA Scientific Speaker Series and a Career Series. They've ended for this year, but they will be continuing in February, so keep your eye on the bioimagingnorthamerica.org/activities page. That's where most of this content will be shared. We'll also have a save the date page up in the new year for the BINA 2024 Community Congress. Please mark the dates. It's going to be in Madison, Wisconsin. We hope many of you can join us. We also have an exchange of experience virtual group that will reconvene in January. We've got 2 other user groups focused on expansion and Lattice Lightsheet that will, expansion has another meeting next week, and then Lattice will reconvene in February. Ooh. And then if you are attending ASCB, likely not, because you are probably traveling. Most folks are traveling there today. Nikki will be leaving us partway through to do her travels there. We're hosting a session on Sunday, December 3rd, and there will also be a roundtable lunch hosted by our corporate partners working group. So if you're there, please do share it. And then Bioimaging North America, the website here, has more information. Again, that can be accessed from our activities page. Just a calendar view of some stuff coming up. I guess December is almost over, so we're working on getting January and February ready. And with that, I will pass it to Nikki. I'm Vanessa. I always forget to introduce myself. I am the program administrator for BINA, and I'm who sends you emails about these meetings. Nikki, go ahead.
Thanks. Yes, so I'm Nikki Biley. I'm the program coordinator, and this slide is really just to remind everybody in this space that by registering for this, you don't necessarily automatically sign up as a BINA member and get the nice monthly newsletter with all the information that Vanessa sends. And even if you are not in North America, we still encourage you to become BINA members. We work a lot with a lot of our international partner networks, and it's a way of finding out about some of the funded opportunities that we provide. So as I say, I coordinate the various programs that BINA has around professional development, exchange of experience, and it's a good way to hear about how we can help you, as well as you participating in this and contributing to the efforts that we have ongoing. So at that, I will hand the floor back over to Judith for intros.
All right, thank you very much, Nikki and Vanessa. So today we have 2 groups of people. One group from Germany, Tom Boissonnet and Christian Schmidt. They'll be describing to us, a brief introduction to i3D Bio from Christian, and then Tom will present on his strategies for Homero annotation. And then after that, we're going to have a presentation from them and a discussion period. And after that, we have another group from the Laboratory of System Pharmacology at Harvard Medical School, Sarah Arena and Alison Meyer. And they will be presenting on cyclic immunofluorescence data and metadata management. And it will be the same format, a presentation and question and answer at the end. So to all the presenters today, thank you very, very much for sharing your knowledge and experience with data annotation and image management. Thank you. So I think Christian will take.
Yeah, thank you. I
now.
just take over. Thank you very much for the introduction, having us here. You should see my screen already. My name is Christian. I will have the smallest part in here today because I'm working as a project manager. I myself work at the German Cancer Research Center in Heidelberg in Germany. And what you see here already is the context of the project in which the work that is presented today by Tom is partly done. And this is the project with this tricky acronym I3D Bio, which is short for Information Infrastructure for Bioimage Data. And what we do in this project, among different topics, is very largely to focus on facilitating Omero-based research data management here in Germany at different universities and research institutes. Sorry for the typo there. And what we do is that we collect material that is also basic introductions to bioimage data. You find this on our website as a little bit of a beginner, really beginner targeted curation of information. And it's a project of 4 institutions here in Germany, very heavily supported by the German Bioimaging Network. And what we do here is, now I go one, yeah, here it is. We also want to support the use case partners with who we implement Omero instances in the training for Omero users. And one thing that we very recently published is this set of slideshows. It's really just PowerPoint slides, but everything is in a very neutral format, and there are markings in there so that you can download the original slides and just adapt it to local instances, reuse it for either training at your institution in lectures or for self-guided instruction manuals. And when you follow the link, then you also find the link to a set of YouTube videos that give just very short videos, step-by-step instructions for beginners, how to use Omero. So we hope that this can be easily reused and save a lot of time on other hands who are beginning with Omero or using it. And that's already the very short introduction from my side. I want to give really Tom time. Tom is our colleague in the project from the Heinrich Hahn University in Düsseldorf, and he's also the administrator of the Omero instance there. He is responsible for helping other institutions in Germany here in the deployment of new Omero instances. And you see the other people here from the universities of Osnabrück, Freiburg, and the German Cancer Research Center. I think many of you know these faces. And with that, I already hand over to Tom for his introduction to his data structuring. Thank you.
Then I will also need to have, if I can share my screen. But thanks a lot, Christian, for the introduction. So that's much less work I have to do now to introduce myself. So thank you. And all right. So what I want to talk about today with you is, well, reflect on how to organize data in Omero, so how I use Omero and how I explain it to my users. And from that experience, yeah, well, give you feedback on my experience, basically. And I'm going to start with how I would introduce Omero at the facility, so at the CAI here, where basically, and I think a lot of you also have experienced that you would introduce all this new concept to a new user, that talking about what is an image in Omero, the data sets and project, the structure to organize the data. We also try to introduce some plugins, so the viewer and Omero figure. We then explain how to import the data, that there's the image metadata that's coming with it. So that's quite a lot of concepts that come on top of that. We start explaining also what annotations are, so tags and tag sets, and the key value pairs. And that's already, well, quite a substantial amount of concept for someone arriving and wanted to do imaging. Trying not to mention too much, well, then presenting this concept, it would also be about linking this concept so that people start to put concepts together, images of course, they're imported and organized with the structures, how you use the metadata to then use the plugins in Omero. There's this other concept arriving on top that we try not to talk about too much at the first introduction, but something coming from behind the publishing, all the electronic notebook management, RMB, image analysis. So these are a lot of concepts to take in. And that would be me introducing all this and saying things. So a year ago, saying things like Omero only has this 2-level hierarchy, but if you need more organization, you can use tags, leaving the users with that. And tags, well, just take this concept from Twitter or Instagram, maybe you know, to describe your data, or that you can use annotation to add metadata. But saying that probably is not convincing enough for the users that would be the reply saying metadata, well, I'll think about how to organize it later. I will take the time eventually sometime in my project and take care of this. And so the key value pairs and tag we have on Omero, then explaining that you can annotate basically all the image, data sets, and projects. The user might actually not make use of it. So because they would have to go through these steps of planning, agreeing on which to use, deciding, so take decisions. And this is something they want to do it only once. And if they actually decide to start using it, will there be pitfalls for them? So ways they would start using it that might discourage them to use them further if they don't see usability in those. And so first issue I would spot is that there's overlapping concepts. So if we take the tags and the key value pairs, describing an image with all the tags, so saying about all the fluorophore you use, which type of cell, which microscope, which condition, those are things you can map back to an extensive annotation of key value pairs. So if you use your key value pairs in the using the RMB recommendations template, then you might end up with a lot of overlap if you tag your images like that. So the users might start questioning, well, why do I use tags if key value pairs do that? Or should I use key value pairs if tags are already talking about that? And so I would try to define now with the users what's the scope of key value pairs and what they should be using it for. Is that with key value pairs, we can really describe in details our data. And so if I take this smaller set of key value pair I use, you could use free key and values like that to describe precisely a term with ontologies, or you could use links to external resources, for example, a protocol in electronic lab notebook, and many other things you could describe. So the key value pairs are very good at describing in details the data. And so that's probably where I would stop saying tags are like hashtags in Twitter and Instagram, that telling about this concept is telling you should describe the content of your data using tags. And this is not something I want anymore. I would prefer to use key value pairs. Another problem I would find, or that I would see the users around me do, is to map the folders habits they have and match them to the project and data sets in Omero. And so, but doing so, they would arrive to quickly a limitation in the structure with Omero that you cannot put more data sets inside data sets. And you would see things like using data set as dump of data, and inside it put all the data from a day of experiment. You could find other structures for those. But when you do this, well, first, how should I be annotating it if I'm wanting to use key value pairs? If I put my annotation on the data set here, I would not really know which image it applies to. I could say, let's annotate the first image of every set, but that might also be unclear on how the annotation relates from one image to another. Or I could be annotating every image, but that could be then more tedious work, or also having duplication of the same annotation. That might also be an issue. And what if now I want to look at the data in some other way? What if I want to reorganize the data to view it differently? What I would see in this data set is that here all these images seem to correspond to a single sample. These images here seems to correspond to a second sample. And so my tendency would be to explain to users that the top part of this data set should be a standalone data set for this sample, and the second part should be another sample. And so basically every sample should get its own data set. And so I would obtain a long list of data set like that for all my samples, for all my experiments. Every time I go to the microscope, I may come back with 3 or 4 more data sets every time. And that increased my list of data for given projects. But now I would like to have some organization. And this is where tags can be useful, that you can use tags just like there are folders. So that's not really possible with dataset. Dataset, again, we cannot put a dataset in a dataset. But if we use a tag to annotate, if we use multiple tags to use tag multiple dataset, then we can find those dataset in the different categories. And I would tell my users to not overdo it. So the tags, again, I don't need to describe all what my data is about. But if I think about them like folders, then I would just tell them to try to map the subfolders they would use on their personal computer to the tag categories here. And if I don't overdo it, then if I have less categories, that's less work of tagging. So that's good. And I might also have less oversight, that if I have less categories, I'm less prone to forget about some categories. So I would also say, if I split my data sets, if I split all my data in separate data set, each for one sample, I would tell them to put the tags on the data set, so that would appear also nicer in this tag menu in Omero. But if I need extra details on the images, or if I need to have more annotation, then I would tell them you can also use tags for images and make special categories for those tags. And so I think this is where tags are good, that they are quite flexible in how you want to organize your data. So really make your folders, and they can match personal preference. So if I'm fine with 3 categories, but my colleagues want to add a 4th categories, then he can add these categories, and that will not disrupt all the organization I had before. So they can add personal preferences to the organization. And so now I can define the scope of the tags, that if the key value pairs take care of describing the data, so I don't need to redo it with the tags. My tags are like folders, but in better, I can search my folders in the order I want. Because I can put multiple tags per object. We can categorize our tags, so the tags are very good for organizing and filtering. I could even try to push the concept and say, let's use the tags to emulate data sets. So I don't even need my data set. I could make tags that would be like data sets. So why do we even have data set structure in Omero? And well, the real good thing about data sets is that we can annotate them. And so I can put tags on data set, key value pairs, attachment, I can rate them, etc. And so the idea would be, if I annotate my data set, implicitly I can annotate what is contained. And by doing so, then I would make less duplication of my annotation. If I put the key value pairs on the data set, then I don't duplicate it on the 10 image it contains. And again, if you need further details for individual images, if there are specificities you need to explain about, you still have the freedom to put annotation, extra annotation on images. And that's why I would also stop say this, that there's only 2 levels of hierarchy, because I would rather provide the way of organizing with tags directly. And instead of generally saying you can use annotation, I would also show this example. So really give those examples to my users. Okay, and when I presented that a few months back to our user group, I would have the feedback that an issue is that adding key value pairs on data sets, well, it doesn't show on the image. So it's probably just a user interface thing. But when I select my image, I don't see my annotation. But so this is work in progress. This is currently an Omero, a pull request in GitHub. That's work I've done to also display annotation coming from a parent. So here, selecting this image, there's annotation on the data set and the project above, and that would now appear in the key value pair. So that's updates, hopefully, that would come and be released soon, but just to show that this is something possible then to display it here. And so there's really good, there's really advantages with that. So if we take use case like high content screening, we would then have all these wells. Inside every well, you would have a lot of images. So putting annotation on every image is very, very tedious. Here I could put again my annotation on the well level. And if I want to, for example, use Omero figure, taking images from different wells, you could also just take, with changes I've made to also add key value pairs coming from parent objects. You could also be displaying the condition, the experimental status annotation coming from the well. So really to give this usability of putting annotation on the parent objects. And so I'm coming to my second part, but really rather short compared to the first one. And I want to talk about annotation tool. And so this is, I think, the purpose of this meeting. And the first one I want to talk about is really Omero Web. So I think overall Omero Web is very strong to annotate data. So it's already has a lot of functionalities. And so I would be importing a new data set, give it some useful name that I can recognize it. And I can already go to a previous annotated data set, select all the key value pairs, select my new data set, and paste the annotation. And like that, I would duplicate all the annotation, put it to my new data sets. I can adapt the keys and values that differs, but that would be a workflow, I think, enough for most of my users. But let's say we have now more complex use cases that would require more tools. And so if you put yourself in a position where you have too many data sets to annotate, so maybe you have not done your work of annotating your data set slowly, or you come to Omero with a lot of data sets, or maybe you're a person doing high content screening. So that's probably just like situation one where you put yourself in that situation. Then we need to have these extra tools. And we have some solution that exists on how to annotate by batch and how more powerful tools to help you annotate. But I think the complexity of this project, or because you haven't been doing your work of annotating step by step, then you will need extra efforts to use these tools. So I think that's where the tools presented in this meeting are very useful. But I also want to mention other tools I've been working on in the past weeks. So on the CSV scripts in Omero, that's for exporting from CSV or importing metadata from CSV, deleting key value pairs, or converting also namespace. And this is also in the state of, current work on GitHub, so not published yet, but if you're attentive again on ImageSC, you will see this work coming hopefully in the next week. But just to show you how that would work, here I'm selecting a plate. And for that plate, I would tell it to export all the wells annotation. There's no annotation yet on my well. So when I do this, I obtain a blank CSV file, but with all the names and IDs of the well object inside this plate that I could fill up in Excel. So just using Excel is a very powerful tool to make batch of filling cells. And I could here, for example, put all my condition for my well. and this CSV selecting back my plate and telling that for every well of this plate, I would like to add the key value pairs coming from that CSV. And so you can also go check today, well, those scripts, they're on GitHub in this working branch. So you can try them and give us feedback. We'd also like to have your feedback on those. But yes, still work in progress. But so to conclude, I have this take-home message that my feeling is that Homero gives you a lot of freedom on how to organize your data. But with all this freedom you have on how to use it, that can be also a lot of decision to take on how to use it. And that can be, I think, intimidating for new users. And so that's why I think providing guidance on how to use it, how to organize, give specific tips on how to organize will help newcomers. And maybe there's a need to develop community standards on how to use it, say that we can organize in this way, and that could be organization level A, and that means something that we can try to then apply analysis to such level of organization or find different recommendations for different use case. And my second take-home message is that Homero Web is a powerful tool, and I think we should try to use it as much as possible before going to third-party solution that requires us to take the data out of Homero and have more complex workflows. If we can use Homero Web, then we should just try to use it as much as possible, and that's probably less overhead of things to learn for newcomers. And with that, I would like to thanks everyone in my facility and everyone from the i3D Bio team, and thank you all for your attention.
Wow, thank you very much, Tom. That's really, really useful as far as I'm concerned. And I do want to comment, Christian and Tom, that what you've put out on YouTube as a playlist in your slide, it's really well done and very, very useful. So if people have not had a look at it, please go have a look, because it's a fantastic resource that you've provided. So I'd like to start with the period of question. And I see that people have already posted questions in the chat. So Ken, do you want to repeat your question to everybody?
Oh yeah, thank you very much, Tom. Great to see the work that you do. Question about the inheritance. So the tags got inherited from the data set. Is it just for display on the Homero web, or is it actually you automatically create a link to the annotation data? And so if you move the image, does the old data set annotation carry on with it, or automatically get deleted and then relink to the new data set?
So it's really just a display. So it's saying if an image is there, then you display the annotation from the parent.
It's a new way to do a Homero display on the web, such that you can see it. Presumably on the insides and other things, then you would not be able to see that.
So yeah, I only made the changes in the Homero Web HTML. Yeah, and then from figure also, that when you're putting a label from key value pairs, that it also grabs the annotation of the parent containers.
So when if you do search of the annotation on your parent data sets, will the images come out or not?
So in the search, it's not defined. I think it could be added in the search to have parameters to say, show me the image with the
as well as the data set.
Yes,
okay,
but so you would have the data set showing up. That then contain, so you could move to the data set.
Images, actually. Yeah. Okay, thanks. Cheers.
Okay, we have another question in the chat from Alexander Smirnov. Do you want to ask your question?
Yes,
directly?
this may have been, hi, it's me talking with a mask. I think you covered it, but so is it feasible that we have some tags or key pairs displayed on images as we like scroll through the whole collection of images?
So I'm
I
not
think
sure
you
what you
mentioned
mean.
that this is not probably not possible yet, but and like just as a text maybe in some corner.
So to put the tag on top of the image.
Yeah.
So like a
so
hover over function, if I understand you right.
Yes.
So I mean, everything is possible if you modify the web page and to display it on top of the image as text. So the Homero web doesn't offer that. That's something you can do in Homero Figure if it's about making a final figure that you can also add tags on top of your images, so as text, as labels, but not in the Homero web display.
And Homero Figure, is that a free plugin? Or is
Yes.
It... Okay.
Yes.
Thanks. And just out of curiosity, like the release of Homero that you work with, is it like a free open source release, or is it the one that's maintained by some companies that I know?
Yeah, by Glencoe you mean? No, so
yeah,
this is
Glencoe,
Homero
yeah.
Web. So yeah, Homero Web is open source, and that's a release on that would be in maybe the next version or the one after that of Homero Web, which is open source. Yeah.
Thank you.
I see. Thank you for sharing your question, Alexander. Ricardo, I see you have your hand up.
Yeah. Hi, Tom. Yeah, thank you a lot for the nice presentation. It's nice because I saw a lot of parallelism. So a problem that I have, I saw that other people have it. So it's really nice. I have a question. So I'm really interested in this part. So this thing that you're developing, that the metadata that you attach to the data set, then it's possible to see them in the image. My question here is, normally if you export the image as a normative in the XML header, normally you can see the part of the key value pair. So if you attach some key value pair in the image and you export a normative, then you will see it in XML. This is still possible to do it, also if you don't directly attach keep a value pair to the image, but to the data set. And then, so with this new method, the metadata will be kept in the XML header or not?
So I would assume that not.
Okay.
But then again, that would be an implementation method. So if at the moment of export, Homero queries the annotation that are attached to the image, you could as well add in your query to say, give me also key value pairs from the parent objects to add them on top in your XML. But so that would be modifying the different plugins that they could also. So what I did is an addition in the API to be able to retrieve also annotation from parents. And so I made modification in Figure that it would work in Figure, but then you would have to change the different plugins that it also gets this new feature of inheritance in a way. So for the export, I think the Mapper plugin could have this in the Homero tag search, the Homero search itself. So yeah.
Okay. Thank you a lot.
All right. Thank you very much for sharing your thoughts. Any more questions for Tom? Okay, so I think we've covered everything. Thank you very much.
Yeah, and thank you for your questions.
Yes, I have more time, but for the sake of time, I'll be back on another time. So again, so if for the sake of time now, I would like to switch to our other presenters. We have Sarah and Allison from Harvard Medical School, and they'll be presenting how they tackle data management and metadata management. Sarah and Allison, thank you.
Hey everyone, so thank you for having us today. So as Judah said, our names are Allison and Sarah. We are from the Laboratory of Systems Pharmacology at Harvard Medical School, and we're just going to walk through how we manage tissue image metadata at the LSP, with a specific focus on our cyclic immunofluorescence image data. So first we're going to go through a little bit about the Laboratory of Systems Pharmacology, what we do as data managers there, and a little overview about what cyclic immunofluorescence data is, because that is by far the most common amount of data that we work with. Then we'll get into the MIDI metadata schema, which is the standard that we use for the cyclic immunofluorescence metadata. And then we'll talk about how we capture this and other metadata within our laboratory information management systems. So a little overview is that the Laboratory of Systems Pharmacology, or the LSP, aims to advance therapeutic and diagnostic research by studying the fundamental science behind them. So we investigate disease biology, we use a systems biology approach, and we're in a highly collaborative team science environment. We study the molecular causes of disease, the therapeutic and adverse effects of drugs, and we apply artificial intelligence and machine learning to make the process of drug development faster and more effective. So this basically looks like a multi-institutional lab. We're a little bit unusual. We work with about 46 affiliated PIs. They're located at other Harvard labs, local hospitals, universities across Boston. And we have collaborators that are located from Oregon to Germany to South Africa. So we're fairly large. Many of the students, postdocs, and lab techs will actually come and they'll sit in our lab spaces at HMS. And through all these collaborations, last year we published about 54 papers. We're highly interdisciplinary, so we have molecular and systems biologists, we have computational biologists, we have software engineers, oncologists, and pathologists who all bring their expertise together so that we can learn about disease progression and response to treatment with the collective goal of making better decisions in the clinic itself. So we investigate disease biology with a focus on cancer, tuberculosis, and Alzheimer's disease. And we use a diverse variety of methods, including highly multiplexed imaging, which is what cyclic immunofluorescence data is under, proteomics, live cell microscopy, and functional genomics. And so because we're such a large group, we do have a dedicated data management team within the lab. This consists of Sarah and myself. We both have a background in library and information science, and it also includes our director of software engineering, Jeremy Mulick. So together we manage about a petabyte of data. We also teach data management best practices within the lab, and we collaborate on lab-wide tools like our laboratory information management systems. And as I mentioned before, by far the most of the data we work with is cyclic immunofluorescence, or CISIF data, which my colleague Sarah is now going to walk you through.
Great. So cyclic immunofluorescence, or CISIF, is a highly multiplexed tissue imaging method developed by Jerry Lin at the LSP. And so CISIF produces really high resolution images, generally at a low cost. So I'm going to play some videos so you can see these. Reagents are applied to a whole slide of mouse or human tissue and labeled with a different fluorophore. We then stain with DNA dye and image the tissue. And then we bleach the tissue to strip the antibodies in order to repeat these steps again with new antibodies. Typically, this is repeated between 8 and 20 times. And each of these images are then stitched together to create an image of the whole slide, tissue slide over each cycle, and then single cell information is derived from these images. This can help augment traditional histopathological diagnosis of disease, helps us learn about tumor immunology and the impact of drugs on cell states. So to create a whole slide image, single image tiles are aligned and stitched to create a whole slide image. Tiles are stitched together per cycle, and then all the cycle images are registered to create a composite image containing information from 30 to 60 antibodies. And I'm just also including the sizes here just to give a sense that a two-dimensional CISIF data set could be somewhere between 5 and 20 terabytes. So as the LSP developed the CISIF method, we also have needed to establish and adapt data and metadata standards to describe, share, reuse, preserve these images. And we worked on establishing these guidelines alongside other members of the tissue imaging community as part of the Human Tumor Atlas Network. So what resulted was MIGHTY, which stands for Minimum Information About a Tissue Imaging Experiment. MIGHTY is described in a paper published in March of 2022 in Nature Methods, and the paper defines the first version of the metadata schema and also establishes data levels, which I'll describe in a few slides. So since highly multiplexed tissue imaging can use various methods to combine spatial and single cell information, MIGHTY is designed to be compatible with a number of image types. This includes images based on H&E staining, high and low-plex immunofluorescence, and IHC. And it draws on existing formats and tools, including OMETF6 and bioformats. So the implementation and maintenance of MIGHTY is a community effort. MIGHTY definitions are recorded on a public GitHub repository in YAML-based files. YAML was selected because it's both human and machine readable. And one of the benefits of using GitHub is to encourage community engagement. So users are able to ask questions and suggest updates to the schema via GitHub, and then members of the MIGHTY governance board monitor and discuss community input. This is still a small group, and we're actively looking to build the governance board and the community. So if you're interested in learning more, getting involved, please reach out. MIGHTY defines clinical biospecimen reagent, instrument, and analysis metadata. The schema is presented as tables with a flat list of fields, and the schema currently provides a name, description, data type, and whether a field is required or recommended. And we also include some controlled vocabularies, but these are all kind of actively things that we're working on updating. So for the clinical and biospecimen tables, we drew upon already established standards such as the Genomic Data Commons data model and the NCI's common data elements. So clinical information includes demographics, diagnosis, exposures, treatment, and follow-up. And then the clinical and biospecimen metadata are linked to all other levels of metadata via participant and biospecimen ID. And because MIGHTY is a product of the Human Tumor Atlas Network, clinical and biospecimen metadata fields currently focus on cancer, but we're working to update these tables to be more generalizable for more normal and diseased tissues. Then table 4, channel level metadata describes antibody and reagents used per channel and cycle in the image represented. And then to describe the rest of the metadata tables, I'm going to return to the steps of image assembly that I was talking about previously. But basically, file.yaml corresponds with raw images before processing. OMETF header corresponds with assembled images, and so on. So this is another view of each step of image assembly, which MIGHTY organizes around the concepts of data levels. Each data level in this figure corresponds to the extent of image processing and lists potential file formats. So each of these levels, and then each of these levels has a corresponding list of metadata. So walking through this figure from left to right, we have level 1 data, which are primary image tiles. In MIGHTY, file.yaml corresponds with data level 1, raw images before pre-processing, and some of the attributes describe the individual file and imaging method used. Then level 2 are stitched image mosaics assembled into one large multi-channel OMETF. Level 3 images include image mosaics after quality control and after artifacts are removed, and then channels that don't look good are rejected. So segmentation, and then segmentation masks, which identify and outline particular biological features, also fall into this category. And so in the MIDI table, this is the process data table. And so quality control metadata describes steps taken to address pixel level issues, and segmentation mask metadata can describe multiple masks per image. Then level 4 data are where cells and features are counted to produce counts tables, spatial features tables, or feature observation matrices. And this corresponds with the cell level attribute table in MIDI. And then level 5 are results derived from spatial features tables or images, as well as human annotated images, which can be presented in various image viewers. And this corresponds with the cell state attributes table, which describes cell type annotations, methods, and softwares used. This is just a list of the required and recommended fields in MIDI. So MIDI is comprised of 199 fields. So that's definitely isn't kind of an active discussion of what are required fields, what are recommended fields, and how do you make this feasible to implement as a minimum information standard. And then finally, there are a number of directions for MIDI moving forward, which include expanding the MIDI schema itself and integrating and exposing MIDI for various publications and repositories. And so some ideas for expanding MIDI include generalizing clinical and biospecimen fields to be more broadly useful for normal and disease tissue types, continuing to build tools for efficient metadata entry, metadata extraction from image files, and MIDI file validation. We've talked about outlining how to handle TMAs and many-to-many relationships between patients and biospecimen. And this could also be useful for integrating emerging technologies like spatial transcriptomics into analysis. We want to add more comprehensive mouse metadata or support for other species. We've talked about creating a checklist for journals to use for multiplex tissue imaging data. And then finally, we are continuing to build out our own tools in the LSP to record MIDI metadata, which Allison will talk about next.
Yes, so to track MIDI metadata as well as other metadata in our lab, we use our laboratory information management systems, of which there are two, Experiment Tracker and Reagent Tracker. These systems, which are actually a single Postgres database on the back end, were developed by members of the Laboratory of Systems Pharmacology in collaboration with the ICCB Longwood Screening Group, which is right around us in Boston. And they were first launched in 2020. They're both open source and web-based. Excuse me. And so to walk you through a little bit about each as an overview, Experiment Tracker is where we track metadata for projects, experiments, slides, and other biospecimen, the antibodies panels that are used within each slice of experiment, and some of our metadata about microscopes. Experiment Tracker was set up with the specific goal of tracking SciSif data, so that is what it tends to focus on. We use Experiment Tracker to issue unique IDs, particularly for projects, experiments, and biospecimens, and as well as helping to manage the LSP slide onboarding workflow. Reagent Tracker, on the other hand, is where we track metadata for two types of reagents. We have small molecules and we have antibodies. So I can walk you through a little bit about the project and experiment metadata itself. If you want to switch to the next slide, Sarah. Thank you. So Experiment Tracker is set up with a project experiment hierarchy. So a project is defined as like a higher level association of particular slides and also the experiments that are performed with those slides. So metadata for a project is going to include lots of like very overview information, like which PIs are involved in the project, what's the funding source, those kinds of things. Experiments, on the other hand, they're more specific. They live inside projects, and they record metadata like the antibody staining panel, some of the microscope settings, some channel information, and which slides were imaged in that particular experiment. And I mentioned that we use the experiment tracker to issue unique IDs. We also then use those IDs. So if you can see, like in the example project, that's project 172, we'll use those as part of folder and file names on our file servers, so to help maintain like clear provenance throughout the whole like data analysis pipeline. And the fields and features that we have are ever evolving based on feedback from users, because it's like an internal database, and we have the people who like coded it in our lab. It's we're always trying to see like how we can improve it. And next I'll talk about clinical and biospecimen metadata specifically with an experiment tracker, because our clinical and our biospecimen metadata is gathered by our clinical research coordinator. And this is a big part of where Mighty comes in, because, excuse me, there are at least 71 metadata fields for tissue imaging, clinical and biospecimen metadata from Mighty that we've asked our CRC to collect. And for internal project, this often means that she's reading through pathology reports and then translating them into Mighty compliant vocabulary. For external collaborators, sometimes it means like getting the information from them. So we've done a lot of work to try and make that also Mighty compliant. What our current system is we provide them with a structured Google sheet, they fill it out, and then we can upload that into Experiment Tracker. The CRC is responsible for managing like our IRB protocols and ensuring that we're compliant with all of our HIPAA regulations, our IRB and the GDPR, to not share any protected health information. So throughout this whole process, she is removing dates, patient IDs, any other sort of identifying information, so that the information that lives in Experiment Tracker is de-identified and can be shared with all of our lab members. Also, we had previously used REDCap to record some of this information, but we found that it wasn't sufficient to record the hierarchy between patient and associated tissue blocks, as well as the sections that are cut from those blocks. So that's one of the reasons we are bringing the clinical and the biospecimen metadata into Experiment Tracker, and are working on that hierarchy. And then the other database that I mentioned, Reagent Tracker. So that is, we store information about two types of records. It's small molecules and antibodies. A small molecule record is going to consolidate information from vendors like MedChem Express, it's Chembl, PubChem, CAS, all of that kind of information, and also some logistics information about how much powder is going to be dissolved. Whereas an antibody record, it's going to consolidate information from vendors like Cell Signaling or Abcam. It's going to bring like the RRIDs from the antibody registry. It's going to bring stuff from Uniprot, and also it records some internal information, such as like validation testing that we've done. And then some future directions for our laboratory information management systems is we are working on further refining our clinical and our biospecimen metadata fields and the structure in which we record them, particularly working on that hierarchy of the patients to their associated biospecimens. We're working on standardizing antibody targets and specifically their purposes, so we can provide that information as context for like when we share images. We want to coordinate the antibody validation documentation workflow, so we record that information better. We would love to expand Experiment Tracker to include other assays, not just CISIF. And we are also working on setting up a little bit of like financial tracking, specifically using the statistics around like projects and experiments and how many slides were used. And then with that, I would like to thank just some of the people who have been involved in developing these methods and these tools that both Sarah and I described today, and who generate and help manage data at the LSP, particularly like to highlight Peter Sorger and Laura Malashevsky's leadership in this endeavor. And then if you want to contact Sarah and I, those are our emails. And we also put up both our lab websites and the website for Mighty. And you can also find Mighty, as Sarah mentioned, on GitHub. So yeah, please reach out.
Wow. That is amazing. I don't know if I want to smile or cry. You have worked out this is just amazing. So thank you so much for sharing. So can you confirm to me that most of these tools are available as open source tools, right?
Yes, so Mighty is available on GitHub. So the laboratory information management systems, they're based on open source code. We haven't, because they're like internal resources, we haven't like packaged them and put them out the way we've defined them yet. We've talked about it before. I just think we haven't gotten around to it yet.
That is really amazing.
But we know that because there's a need for us, there's probably a need for other people. And so it's definitely a conversation we've had.
Yes.
Mm-hmm.
So, all right. So let's move on now to the question period. So I see Alexander has his hand up. Alex.
Yes, thank you. Wonderful efforts. And I'm thinking that will be a great help when some of us may start applying the same things, like for neuroscience, for example, and for actually starting from animal models that you plan to work on, and then maybe extending later into clinics. I just kind of have maybe more technical question on when you overlap, when you go through different stages of images, I imagine from each cycle, the images not perfectly overlap onto each other. And how do you manage that? I mean, do you keep all the raw data, or do you somehow morph different images together? And maybe there are some regions that gotten bad during the process of cycling regions, and you mask them out. How are you address those kind of artifacts that come during processing, both physical processing and computational processing, when you stitch them all together?
So I know I don't know if I'm gonna say that would be a question for our software team, unless Sarah has anything she would like to chip in about that. I'm not particularly involved in a lot of the specifics with like how we stitch the images, so I'm not exactly sure how we deal with like the overlapping and where the stitching actually happens. I don't know if you have anything, Sarah, feel free to jump in.
No, we have MC Micro is the analysis pipeline we use that it handles a lot of kind of the routine things that need to happen to like QC and align images. But so yeah, but if you want to email us, we can also pass this question around and get back to you with more concrete answers.
Oh, okay, yeah. So sorry, more relevant question is, so you used limbs that you developed in-house, and these days there is like hundreds of ELNs and limbs that's like already pretty good. I'm just curious, like, have you explored some of them, and what you found that encouraged you to like do it yourself, really?
Yeah, so we also do offer our lab members an ELN to record information about their particular experiments. I wasn't involved when this, like at the very beginning of this project, but a lot of this was around the issue specifically of capturing the like reagent information and antibody staining panels. And so I think those particular needs couldn't be addressed by the ELNs that were available in 2020. And so that's kind of how this project started, was around the particular issue of staining plans. Allison, do you have more information?
Not particularly, just that kind of like the staining plan kind of being the heart of Experiment Tracker and what it grew out of is definitely very true. I think it's also could have something to do with the fact that because we are a very interdisciplinary lab and we have like software people in the lab, it was a very like, oh, we can do this ourselves and make it as customizable as we want it to be. So maybe that was a bit too tempting for us at the time. I also wasn't around at the beginning of these talks, but that's kind of my what I get from the stories I've heard.
Thanks, fair enough. So good to have capable people around. Yeah, of course.
Yeah, it makes a huge difference, and we can see how we can move forward in such an elegant manner when we have the resource and the qualified people for doing this. Thank you. So I see Z has his hand on.
Yes, great stuff. I would very much encourage you to get it out on GitHub as soon as it's ready. When we were looking for stuff in 2020, there was nothing on the ELN side that would give us the flexibility. They get it almost 70-80% all the way, but that remaining 20% was something that would actually break everything. My question to the team is, are you working on supporting other types of experiments, such as, for example, the things that Orion Rare Site is doing? I noticed that you've using their microscopes for the cyclic IF, and I was wondering if that's also part of the plan.
It is. So we're currently working on being able to record the way that the panel is for an Orion experiment. We have recorded, right now we kind of have a half solution where we've recorded most of it, and we're working on updating a little bit of how the panel is actually structured to be able to also record like Orion experiments as close to, ground truth as possible.
Great. Oh, what are the ways how you can export from this database into some kind of other file type? Do you support like CSV export, and how does it look?
So do you want to take it?
Sure. Yeah. So that was an important feature that we realized we needed, was being able to export metadata, especially for as part of our work with the Human Tumor Atlas Network. There's pretty extensive metadata required, and so being able to export, and now we're also working on importing Excel and CSVs in different formats. Basically, we got to specify which format is needed for different situations.
Mm-hmm.
Yeah, I think when we are starting to upload our data to repositories for the NIH requirements, we often are like, yeah, the data are already here. And then we come to the metadata page and there's like, oh, wow, this is painful. So if someone could just wave their magical coding hand and create something like, this is your format, spit out to a format that's gonna be acceptable by Zenod or anything like that. That would be fantastic.
We have big challenges ahead of us, that's for sure. Thank you for the question. I can see now Steve has his hand up. Steve, please.
Yeah, thanks, Judith. So my question is about the image data. Do you have the image data actually in a database, and is it connected then to this experimental metadata LIMS system that you have? Or how do you work that?
Yep. So yeah, it's not within the LIMS system themselves. We keep our data on the file servers. We've talked about how to connect it. We've had we've had different discussions about whether we should connect, just have the path within an experiment of like, this is where the data lives on the servers. We've gone back and forth about that. How we do it right now is that we use those like a project number or an experiment number to be able to organize the data. So all on our file servers, all of our SciSIF data is organized into projects, and it'll be, project 172 dash some, shortened name that's descriptive. And then under that, you'll have all the experiment numbers. And also every like slide that gets onboarded into the system gets a slide ID number, and that will be used when the slides are imaged, so that hopefully most of the image names for the actual files are going to have that slide ID in them. So that's how we try and keep the connections. That's how we currently do it. But yes, we've also discussed trying to do some other stuff. We just haven't implemented anything on that yet.
And people have also talked about wanting some image within the database, especially for antibody validation, because different people will kind of make different judgments, and so they want to be able to see for themselves. So those are all things that have been talked about but don't currently exist.
Yeah, we've talked about like thumbnails before and having thumbnails in it, because when we onboard slides, sometimes we'll put in like pictures, the CRC will put pictures of the slides that she takes with like her phone, and she'll put it in there. So we have, the actual pictures of the slide batches as they're uploaded. There's also some workflow stuff I think we would have to work out before we did thumbnails for the actual images, about like who uploads it and all that.
Thanks.
Thank you. Alex.
Thank you. I just realized at Johns Hopkins, we had I was not involved in it. It was some time ago, but I wonder if you're aware of this effort called AstroPath, and I shared the link in the chat at Johns Hopkins, where they joined forces from astronomy, and pathology, and added some kind of graph theory on top of that. And it was also, I think, multiplex imaging for cancer, kind of therapy prediction. Any cross pollination there? Or if not, then maybe good idea to try something.
Yeah, thank you for sharing this. I haven't heard of this before.
Yeah, neither have I. Thank you.
Okay.
Yeah, thank you. This looked really cool, and also the Mighty guidelines when they came out. It's really nice that this effort is ongoing so much in depth already. And if I remember it correctly from the paper, and please correct me if I'm wrong, you have also already integrated this, how was it called, Minerva, to visualize the data in a pretty nice way, so that you can really tell stories and so on. And my question is that very much tailored to the structure that you have it? Because it looks like such a cool and powerful tool also to communicate the science to the public, communicate it in papers, for reviewers, for colleagues, for everyone. So how generalizable could Minerva be? For example, to run with Omero, who knows?
Yeah, so Minerva came about in part because our images are so large that there isn't a great way for people to access them outside of an Omero server. And so what Minerva does is basically take the OMIA TIFF and create a series of JPEGs to make it much smaller, and can browse the full resolution story in just a web browser. I know Minerva we've certainly used for 2- and 3-dimensional SciSIF, and for H&E images, and also for MIB data. So I think you do maybe have to convert it to an OMI TIFF in order to use Minerva. And I'm not sure what the plans are if there's plans to make it available for other file formats. But we certainly think Minerva is a very cool tool. and are trying now to, our more recent Minerva stories include metadata from our experiment tracker database, especially around clinical and biospecimen metadata, to try to add some more context to what people are looking at.
Okay.
Thank you.
Thank you. Wow. Look, we already passed the hour and we have some friends over the Atlantic, and they're very generous to stay with us on this Friday late of the end of the day. So I think we are gonna go and move on to our concluding remarks. Thank you so much for sharing this. I want to remind you that it's being recorded and we have a shared folder, and we can personally, I think I'm going to have to go back and re-listen to these presentations, because there was a lot of very important information that was shared and very inspiring. So thank you to the presenters. Thank you to the participant for sharing your thoughts and asking very interesting questions. And I don't know, Vanessa and Nikki, if you want to add something else today.
I think just a final comment of, if you have any folks that you know who are also working in this space who might like to present at future meetings, we do have the team from German Bioimaging. I'm sorry, I've forgotten the project name, but they will be presenting in February. Judith knows it.
Suzanne and Stephanie.
Yes, they'll be presenting on their work on data management. But if you know people who might want to present in April, June might conflict with ELMI, but we'll figure that out and we'll keep you guys all updated going forward. Thank you all so much.
Thank
Happy
you all so
December.
much.
Hard to believe it's already here.
And have great holidays.
Yes,
We will not
safe,
see
happy
each other.
holidays, everyone.
Safe and happy holidays, everyone. All right.
Goodbye, all. Thank you so much for joining us today.
Thank you.
Thank you. Goodbye.
Bye-bye.